Hello, everyone. It is my great pleasure to introduce to you a distinguished scholar and researcher, Dr. Gail Dosset. Dr. Dosset is the director of the Brain Architecture, Imaging, and Cognition Lab at the Institute for Human Neuroscience at Boys Town National Research Hospital. She earned her PhD in cognitive neuroscience, focusing on identifying resting state network and their link to mind wandering in healthy population. She further pursued her training in neuroimaging applied to clinical population as a postdoctoral fellow at Thomas Jefferson University and then at the Icahn School of Medicine. Her lab uses multimodal neuroimaging technique to investigate the impact of age on the brain network architecture through the lifespan and how it impacts cognitive function in healthy and clinical population. Her work has been funded by National Institute of Health. Dr. Dosset, thank you for being here. And without further ado, I let you take it from here. Thank you.
Well, thank you for having me today. It's very exciting. And I thought I'm hoping we can have a good discussion after about brain functional atlases. I think it's a big topic, and we're not discussing enough the pros and cons of it. So I thought I would try to give my point of view on that. So as Armin said, so I'm a cognitive neuroscientist. And when I started like neuroimaging, it was like over 15 years ago. We didn't have brain atlases, and we were like, how do we define brain functional networks, right? And we actually still don't know really. But it was kind of a major question in how many are there and what is their size and how do we compare them? And it was kind of still like really something we didn't really know. And so I think with years, people have started to use brain atlases. And here I'm obviously only talking about functional atlases, so based on typically resting state fMRI. And I think it has been really instrumental in cognitive neuroscience to be using brain atlases to help define the networks and then compare across studies, because that's not just to have atlases, it's how do we compare across studies, and brain atlases has been of great use. However, as you probably all know, there are so many options. I think, let me, we know, I think Kio atlas is among the most common atlas used in the field, or Schaffer. But there are so many, and it's still kind of a question of which one should we use in research. And there is no good answer yet. And maybe someone has a good answer, hoping, but yet it's not there yet. But hopefully, like recently, Lucina Udin, she created this great toolbox, the Network Correspondence Toolbox. I'm not an author, so I'm just advertising for her study. But it's kind of to account for this variability and those atlases. There are so many. How do you define your network based on which atlas? And so this toolbox allows to quantify the overlap between a specific network and multiple networks, atlases that are available. So it's really cool, I think, to use that to help define networks and names of networks, right? Because we also, in addition to not know which atlas we should use, we don't know how to name the networks. So here, for example, they extracted this brain network from, I think, the UK Biobank, and they test this network across 8 atlases available, and then each network within each atlas, and then define which one are the most overlapping. And in this case, they found out that this network is more similar to whatever we call singular opercular network or the salience network, depending on which atlas we're using. But so it can really help define networks when we don't really know what they are. And from this toolbox, they have actually currently 23 atlases, but really 16 unique ones, because you know that a lot of atlases now provide different level of parcellation. For example, Schaffer has 200, 400 parcellations, or the Yeo has been also parcellated in different number of ROI. So you can choose different atlases and parcellation to compare your own networks. So I think it's really cool to kind of start understanding why atlases are different and how we can try to understand and combine them instead of just taking one. We can try to integrate all this information together. However, so while it can really help identify and name specific brain network, it's not designed to explain the large variability across atlases. And we know there is a lot of variability across those. So a few years ago, I was like, hey, which atlas should we use for our study? And I started to list multiple atlases of functional parcellation that were available at the time, and it's really not complete. Please, I apologize for that, but at that time it's what we had, and I think it's more to give like an overall idea. But so I was looking into all those different parcellations, and it's like, if you can see, like the sample size is very variable. The age range is always typically, young adults, we know that. But also the number of networks that are revealed within each parcellation is also largely different. And so it's kind of confusing, and I think a major issue in the field where we don't know what to do of that. And here it's just, again, the visualization of those different atlases, and we can see just, by looking at the qualitative difference between them. So some, include subcortical and cerebellar networks, some do not, some cover the whole brain, some do not. So it's kind of a mess if you think of it. And if we were now adding all those new atlases that have been created since, it would be even worse. But anyway, at that time, we were trying to compare those atlases to try to define something, try to understand which one is better, which one may be more reliable. And so we started to categorize each network of each atlas within 5 large networks that we kind of all agree about on the visual network, the sensory motor network, default network, salience, and central executive network, or frontoparietal network. And so what you see here is the overlap of each type of network across the 6 atlases. And so when it's dark red, means they all agree. Well, if it's blue, it means only one atlas have defined this specific voxel as part of this specific network. And so you can see overall that the visual network was probably the most consistent across atlases, but like the DMN is wildly different across, like there is a lot of blue, meaning very different parcellation of the DMN depending on those atlases. Same thing for the salience or the central executive network. So it's kind of wild, I think, how much variability there is between just 6 atlases. And so we computed a map of same thing, flexibility, where it's basically how much a voxel has been assigned to the same atlas. And so when it's blue, it means it has always been assigned to the same network across the 6 atlases. So in blue, it's very stable, basically. And when it's red, it means it's very variable. It's never assigned to the same network, which is kind of consistent for the subcortical, because we know, for example, the Yeo atlas did not have subcortical, so it's going to be more variable. But the region in blue are mostly located in, medial regions of the brain, especially the DMN is relatively stable in the, you can see the anterior and posterior part were at least relatively stable across atlases, or the primary occipital regions as well as part of the visual network. But they still, it should be darker blue more often, right? They are still on the lateral side of the view. You can see that there is not that much darker blue, which means that there is a lot of flexibility. Again, that a voxel is not always attributed to the same network. And so it's weird, We further compared each pair of of atlases across all the networks together, and what we revealed is that the atlases that were based on larger samples were more similar to each other. For example, Yeo had 1,000 subjects, so and our parcellation of 2018 was based on the HCP young adults. So we had over 400 subjects at that time. And they are the most similar to each other, even though we use very different approach to parcellize the brain. In our case, we used, volume-based approach. Here is a surface-based approach. So one didn't have the subcortical we had. So, and at the end, on the other side, the networks that were the atlases that were the most different were those based on the smaller sample size. So, for example, those two atlases or parcellation were based on less than 30 subjects about that. So, it seems that really the sample size matters a lot in defining in the quality of an atlas and how reliable, maybe, or how comparable it can be from another atlas. And so that's kind of one of the conclusions of this paper was that there is definitely a very strong effect of the number of subjects included in the atlas, but obviously the differences may also be linked to sequence acquisition, preprocessing, and also surface versus volume based. But for those two last parameters, I think it's not as important as we may think, just because we can see that if we have enough subject, we're kind of compensating, and the results seem to be more reliable and more similar to other atlases. So if there is anything, I think we should favor atlases based on larger sample sizes, which I think now it's not an issue, right? Because Yeo or Schaffer Atlas or Gordon, they all have very large sample size. So I think it's really good that the field has moved to really large sample size, at least for brain atlases in that case. But the other issue I think that we're not addressing enough, and I think maybe it's starting to change, but I'm not totally sure, it's the impact of age on the brain network. As I was telling earlier, most of those atlases are based on young adults, right? However, we know that age is significantly impacting functional connectivity, which are the base of brain network. And we know in early life, there is a very strong change of network parcellation and modularity of the brain, as well as in late life. When we get older, the brain network become, more connected or less connected within, and so on. And so how can that impact most of analyses that are done on other populations than young healthy adults? We don't know, because we don't really have a lot of atlases available on other population than just young adult again. For example, ABCD is based on the Gordon Atlas that is based again on young adults. So I think it's time for people to start thinking of doing more. I know Vince has recently published last year the Neuromark 3 that has now an atlas for infant and young children and young adult and late life. So it was really cool to see that, and I think we need more of that. And I will now talk about the two studies that I have on those different populations, just to bring more to the field. And so, Boystown, where I work now, is like a very strong, like a large children's hospital. And so I had like a large project on children and adolescents. And, we keep hearing how puberty and just early life, impact the brain and the brain networks. And it's like, well, we should then have more atlases that are focused on this age group. And so we came up with Dev Atlas, which is based on resting state fMRI from 3 different cohorts that are available in the US, upon request, the PNC, the HCP development, and the ping samples. And so after excluding subject, the R sample is 1,391 healthy kids. And overall, so those were based only on subjects that are between 8 and 17 years old. We also used an independent sample of a little bit over 200 typically developing children that we collected here to compare. And then you can see the distribution of the sample. It's very well distributed between male and females. And we have quite a large sample for each age, each year, between 8 and 18. And we used an approach to a single subject ICA approach to create the atlas. So that's based on an algorithm that was developed during my PhD with my previous lab in France. It's called Mika. It's a multi-scale, so it's a very quick description of how we do it, and it has changed a little bit over the years. But it's basically we run a single subject ICA on each subject, so like each of them multiple times. And then we have some clustering approach by group of subject to check for repetition and reproducibility to make sure a network is found multiple times in each subject and so on. And so from this approach, we found 56 components. And then we kind of exclude subject, components that were non-reproducible across both samples. So that's where we use a second sample to make sure that a component we found in one sample was also in the others, because if you couldn't find it independently, that probably means the network is not really stable and important. And of course, we also excluded any noisy components, white matter, CSF, and so on. And so from that, we were able to identify 24 networks that we classified within 6 systems. It's total, to be honest, it's totally arbitrary. We use a 6, DIFO mode, attention, dorsal attention, control, sensory motor, salience, and visual, just based on, like other people's research, like especially we use the Yale Atlas kind of as a base. To classify those network. And so, but in our case, we used, a volume-based approach. So we do have subcortical and cerebellar activation recruitment, as you can see in many. I think it's quite important, especially in this age group, to include those and to not just stick to the cortex, because we know the subcortical chain connectivity, will change a lot at that age, as well as the cerebellar connections. We also choose to use a non-overlapping approach. So each one voxel is only in one network. We could debate. I know we talked with Vince about that, and it's a point of view, right? I just chose that because it's more common, compared to Schaffer or Yale. But overall, so we and I use the NCT, so the Network Connect Correspondence Toolbox, just to compare with adult atlases. And so we use the first network we found that you may recognize as more part of the DIFO-MOD network, and it is what this toolbox found. You can see that most of the 7 atlases we chose were largely associated with one of the DIFO-MOD network or one of the sub-network from this group. And so that's the overlap between, in blue, it's our network from DevAtlas, and in red, it's the other atlas. And you can see how much variability there is. So I don't want to comment on that. And I know people have asked me, like, oh, what do you think it means? But I don't, because there is so many differences due to, age group, approach used. So it's impossible to know why it's different, as much as it is. Just because we cannot control for everything, so there is just no way to know. After, we still can recognize the DMN, and it's making sense, right? Those kids are still close to adulthood, and they're just human, so we will find the DMN anyway. And then we because we looked at 8 to 17 years old, we further test the effect of age on the functional network connectivity using GIFT. And we did it within the PNC sample and the HCPD. That were the largest samples we have. And that way we didn't have to correct for sites. We did it really at the individual level. And we found a lot of effect of age. So even if it's only, 9 years, we know that this age, again, is associated with many changes in the organization of the brain. And what was, I think, kind of cool. So first, you can see how similar those 2 matrices are. And then, so in yellow, it shows positive association with age. So each, pair of network or functional connectivity between pairs of network. And in blue, those are negative association. And so what we can see is that within each system, we can mostly find increased connectivity as the children get older and more adult-like. But really one of the only networks that shows some negative association is the DMN with the other networks, right? Meaning that as those children become more adolescent and then adult, the DMN become much more specialized and become more and more disconnected from the other network, which is, making total sense, right? It's basically what we know of becoming an adult, and DMN is going to become like really central and more independent from the other networks. So it was kind of good to show that, but that means there is still, even if we already selected a subgroup of subject with a more age restriction, there is still a strong effect of age within this atlas, but not, yeah. We also looked for sex effect because of puberty. We know there will be different between girls and boys, but actually we almost found really very limited differences. So I'm not going to really comment on that. But again, across the two samples we tested, we found very similar results. So blue would be stronger connectivity in female than in males. So we found mostly, in the DMN, or and then we found some increased or higher connectivity in the male than in female in yellow. But again, it's not that much. Here, all the results were FDR corrected. And so I think that's where, we need to investigate more studies using age-appropriate atlases and test the effect of age on developmental sample. It's such a big hot topic right now, understanding development, especially with the ABCD, and yet we don't have enough atlases that are based on this age group. So hopefully, we have now DevAtlas, we have Neuromark, as I was saying, from Vince Calhoun, but we need to, do more and to compare them across each other to see a little bit how they are different, and if they are, I don't know if we can say one is better than another, but we need to have more options, I think, especially in the context of development. And kind of the next plan for us, given the strong effect of age, is to provide now DevAtlas at a smaller age group. So we're currently creating atlases, or at least networks that are based on 2 years age group. So we are like computing networks at 8 to 10 and so on, 10 to 12, and hopefully we can kind of compare the networks across those age group and understand which network will have more changes than others, and make that publicly available. But right now, DevAtlas is, again, it's publicly available. You have like the GitHub. I hope the QR code works, but I'm not totally sure. But otherwise, you can always Google it. The paper got recently published too in DCN. So, feel free to, I hope people can use it to test it. We need to test it on different children population and adolescent population with maybe mental health risk. I think would be really great to see if it can be better predictor, use more sensitive maybe to differences. And I believe it's available on the GIF toolbox, but I have never checked, so I don't know if it got updated or not. And so on the other side of the life, right, aging is another topic of interest in my research. And same thing when I did, like, it's a little bit older actually, this project, but looking into healthy aging, and there was really nothing, no atlas based on older adults. And knowing that there is such a large variability in brain networks across older adults, that also may be something to think about it. And that's not a study of mine, but I will talk about it a little bit more later. It's a study by a Swiss group, and they did a literature search on healthy aging, and they looked what atlases they used. And the large majority of study used the Yale Atlas. Not totally surprising, but it's still there. It's still not based on older adults. And then within the Yale Atlas, they found that most of study are using the 7 network parcellation. But it's kind of very, I think, interesting to see how much variability there is across study, and that there is really no consensus. And so we created a few years ago the atlas called Atlas 55 Plus, which is atlas based on data coming from older adults above the age of 55. And so at that time, sorry, we didn't have HCP aging yet. So we used the CAMCAN sample from Cambridge, the SOLD, or SELD, I don't know how to say it, and the ADNI sample. So we selected everybody above 55 years old. And so we had a sample of 563 subjects with resting state fMRI. So for an average age of 70 years old. And you can see the distribution there. So we use basically a very similar approach that I described for the Dev Atlas about clustering, single subject ICA. And so at that time, I did it slightly differently, where I computed the atlas or the networks within each cohort. And so what we found, we were able to identify networks within each cohort. And then we grouped those networks across only keeping 5 networks, 5 systems, I guess. The DMN, again, Salience, executive control, I keep calling them differently, but it's always this frontoparietal network, visual and sensory motor network. So we had that for each cohort, and then we compared them. So the idea would be that those 3 cohorts that are only based on older adults should be more similar to each other than if you were comparing to younger adult atlas. And we did that. We also compared each network, the spatial distribution for each network, and across those 3 atlases. And so what we found is, as we would expect, those network covering primary cortices, sensory motor and visual, were much more similar across cohort here between the 3 than the salience, for example, that was really low. Surprisingly, I mean, salience has been always, I think, one of the most variable networks. So that's what we found. And then when we compared to a sample of only young adults, so we use a CAMCAN since it was coming from the same scanner than the CAMCAN. The CAMCAN has a sample of like over 600 healthy adults between 18 and 88. So for the Atlas 55, we only use people above 55 years old, and we also computed the same atlases with individual under 35. And so we were able to compare each atlas to the SOLD and ADNI. And what we found, as we would expect, is that when we only use older adult atlases, we found better similarities than when you compare them to an atlas based on younger adults, where we can see the grades always showing a smaller overlap. So that's consistent with what we know, but that's, again, insisting on the fact that we should not choose an atlas based on young adult on older population. But that's something to argue, because I will show another study that actually is not as clear on that topic. Anyway, so at the end, we computed Atlas 55 plus, and we have this 5 network parcellation, but we also have, we also parcellated it into 15 networks. So within each, we also provided like a sub-parcellation. And it's, I think I updated the link, but it's available online as well. Switching. We, I see time is flying, so I will go a little bit faster. But we compared again Atlas 55 to this, the same network in younger adult, and we found that the major differences were, I think it's quite cool, is that, the DMN is typically including the hippocampus, right? But we could only find that in the young adults, in the red network here, while the DMN of the Atlas 55 does not include hippocampus. It actually has been switched to the executive network, so it's quite interesting, I think, and it's kind of consistent with the Desha hypothesis about how the executive network and DMN may be a little bit more connected as we get older, with Nathan Spreng hypothesis. So I think it's really showing that using the DMN from a maybe younger adult atlas may not be really reflecting the true DMN of older atlases. But that's could be a debate on that. I'm open to question, in other interpretations. And so we at that time, we did apply this atlas to investigate cognitive decline. And for that, we use, again, the CAMCAN sample. And the idea was to, we also use the neuropsych data that they provide. They have a really good sample with at least 36 cognitive variables across, executive function, working memory, language, and motor. And so we extracted this information, compare the older versus the younger population. So that's just the effect size that I will skip now, but we found typical finding where older adults show, a little bit lower scores than younger adults and slower reaction time. But from those data, instead of just comparing, IQ, we computed a score, an index called person-based similarity index that we developed. So the idea is to compare the cognitive profile of one individual with the cognitive profile of other individuals. And so you would have, you're basically, instead of doing intra-subject correlation, it's like an inter-subject correlation. And so the idea is that you can compare each subject to each other, like each pair of subject, and you get a score per subject, which reflect how similar this subject is from everybody else. And so in that case, we compared each older adult to, so the cognitive profile of each older adult to the cognitive profile of younger adults. And so a high score would suggest that their cognitive profile remains very similar to younger adults in the way that they are probably keeping higher, they have higher scores across the board. If they have a lower PBSI score, that would suggest they are more different, and so maybe more cognitive decline. We confirmed that there was an effect of age on this score, where it's what we would expect, that younger old adults have a more similar profile to younger adults, while as they get older, this profile becomes less and less similar. And so the idea after that was to see whether we could predict this score using structural and functional MRI. And so first we just extracted typical, subcortical volume and cortical thickness and see if we could predict this PBSI score. And it was actually quite bad. We only could explain 6% of the variance. And when we used Atlas 55+, so we extracted the connectivity within and between network for the 15 network parcellation, we were able to predict 17% of the variance, and it was beyond age, sex, and education. And so I think that's kind of promising, saying that this atlas can be used at least in older population to try to predict cognitive scores. It seems to be at least sensitive. In that case, we didn't compare it to any other atlas, so I cannot say if it's better or worse. But at least the results were very significant, and it was interesting that we saw that most of the effects were driven by networks of primary cortices versus in the sensory motor versus executive networks. So as those networks become more disconnected, the older adult had better scores. So that's what we expect. Basically, that's what we were expecting. We were hoping, at least. But yeah, so overall, Atlas 55+ has been adapted to older population, and it seems, at least based on our first study, that it may be sensitive to age-related cognitive changes in late adulthood. So I thought it was good, but we need to do, again, more studies using those different atlases and compare them. And that's actually what this very recent study did. So I'm, again, not totally independent of this study. And you will. So it's a group in Switzerland, and they wanted to compare the Yale Atlas to Atlas 55+ in their sample. And so they have a really impressive sample, actually. So it's a longitudinal study with over 200 participants, and each participant has up to 5 data points collected over 7 years. And so they extracted the 5 major networks, and for each parcellation, so 5 parcellation at A55 for Atlas 55, and then that the Yale Atlas with 7 network, 100 ROI, 200, 400, And somehow they also did 17 network parcellation with 300 ROI. And so they just compared them, and they aim to, so they did multiple analyses. So their main analysis, and I think that's where I a little bit disagree after with their results, or the goal of their study was really focused on investigating the effect of age. And so for that, they use a latent growth curve to see whether there was a linear or nonlinear effect of age on their sample, and the degree of variability between individual. And then they also computed two measures of homogeneity, kind of on how which atlas better suits their data, that they call so regional homogeneity and a silhouette coefficient. And it's kind of a, it's like across time as well. So it's not just like the regular ReHo that we know from resting state, because they also included time in their analysis. So it was a little bit different. But overall, what they found is that, and I will obviously not go over all the results, because it's not the point. It's more about, again, opening a discussion, I think, about what atlases we should use or not. But I think what I pointed here at the differences were, so they found that depending on the atlas and the level of parcellation, or even the Yeo atlas, like that's where I think it's interesting, is that even within the Yeo, they didn't find always the same result of linear versus non-linear effect of age. But they also found, that using Atlas 55, we found a linear effect, while with the Yeo, they found, for example, a non-linear effect for the salience network. And so they found some differences, but I would say still for most of those networks, within each network, they found very similar results. So that's kind of good still. I think it's not totally different across the board, so it was good. So those are the results of the ReHo and silhouette coefficient. And so just to, I'm not gonna go into detail, but basically what they're saying is when the P is significant under 0.05, it means that in theory, the atlas is better fitting their data. So in that case, we see a lot of non-significant result, meaning that it doesn't fit well. But otherwise, what they describe in their result is that the Yeo seem to be a better fit for their own data, because it's more significant. Although it's true that the Atlas 55 doesn't seem to be very significant compared to the Yeo, but I'm still not totally convinced about that. Anyway, so I think, and I'm gonna end here. But the conclusion, I think, and that's what also they wrote, so that's from their paper, and I totally agree with that. I think we need more studies that use and compare atlases with better QC measure, because I think that so far, when we found good result with one atlas, we try to find an atlas that is the most similar to find similar results and say, hey, our results are reproducible with the second atlas. Yet we don't use QC measures. So I think it remains very, the interpretation can be also discussed, like, what does that mean? And most of studies anyway only use one atlas, and we just generalized to all the atlases, but it's not the case. So I think it's something to consider. But I think really the goal, and I think that's where I disagree with what the previous, like the Swiss study did, is that they didn't have any cognitive variables. And I think that's the main error. And they do discuss that a little bit, that they explain that why we found less effect of age on the atlas with the Atlas 55 is because we already corrected for it in some way. So I really think that depending on the purpose of the analysis that we plan to do on a specific sample, a different atlas may not, we should think of which atlas fits better. And so, yeah, I think Atlas 55 or Dev Atlas would be more useful, I think, when we study cognitive variables on this age group, because we correct for age, we kind of, and so the point is not to study age with those atlases, it's to study something else. And so that's where we should think about. But again, we should really try to validate which approach we use, which atlas we use, with behavioral outcome. I think that's a main point. But I'm happy to discuss it more with you guys. And so I just want to thank everybody for listening to me and my team at Boys Town and all the NIH grants I received. Thank you very much.